Phytochem Talks schedule here. Today we have Monica who will speak, Monica Scognamiglio. She'll speak about NMR-based metabolomics, and I'll talk more about that in a second. But you can see here the rest of the schedule. We've got Eliana Gonzalez-Vigil will be with us in 2 weeks, and then our own Lars Cruz a couple weeks after that, and then Marco Todesco in December to wrap up the fall 2022 schedule. Please remember that we have a website. This is phytochemtalks.github.io. If you Google Phytochem Talks, at least for me, it's the first hit. Please feel free to share that link with your friends and colleagues. You can sign up to be on the mailing list there, or they can. And it's also possible to nominate speakers for future sessions. There's a nominate button at the top, and you can always nominate yourself as well if you wish. All right, so as I mentioned, today's speaker, we have Monica Scognamiglio with us. She received her PhD in 2011 from the Second University of Naples with Professor Pietro Monaco. She's had a variety of postdoctoral positions and was an Alexander von Humboldt Fellow in the NMR biosynthesis group at the Max Planck Institute for Chemical Ecology. She's currently at the University of Campania, Italy, where she's an assistant professor of organic chemistry in the Department of Environmental, Biological, and Pharmaceutical Sciences and Technologies. And her current research objectives there are to enhance our understanding of why plants produce lineage-specific chemicals, as well as to identify bioactive compounds with applications in medicine and nutrition. She's been an author on more than 75 articles, including some from her own research group now. I saw it just came out in the last year or so. And she has, I first heard about Monica through Twitter and through her blog. She has a great blog about phytochemistry, and you can find that if you Google her name, it'll go to the blog there. I think the most recent post was about resveratrol just a week or so ago. So that's all I have to say. The floor is yours, Monica, and I look forward to hearing about NMR-based metabolomics today. Oh, no, I think you might be muted. I think we can see your screen, but I think you might be muted, Monica.
Yeah, okay. Now you can hear me.
Yeah.
So thank you for your introduction and also for the invitation. So today I will talk a little bit about our work on plant metabolomics. I'm sure I don't need to convince you that plants are excellent chemists. They produce a wider range of specialized metabolites, which are characterized by an enormous structural and functional diversity. Now, when I'm talking about the structural diversity, I'm also talking about the 3D structure, so the stereochemical aspects. This structural diversity results in extraordinary selectivity and specificity. From the plant's perspective, these compounds are involved in interaction with the environment, with other organisms, and of course they are involved in the response to stress. But humankind has been using these compounds for several purposes since a long time, even before knowing about the existence of these chemicals. And somehow these slides also summarizes my research interests. So our group is working on the isolation and structural elucidation of bioactive compounds, but we're also very curious about the role these compounds have in planta. Now, since we know that plants are such great chemists, we also are already able to understand how complex an extract from plants can be. And metabolomics can be very useful to study the chemicals that are produced by plants. It can be used not only to identify bioactive compounds, but also to study their biosynthesis. It can be applied to several aspects of natural product research. However, today I will be mainly showing you the application of metabolomics to bioactive compound discovery. And especially, I will try and convince you how this approach can speed up the discovery of bioactive compounds. Since we are talking about very complex mixture, by combining the information that derived from their chemical composition and the biological activity, using multivariate data analysis approaches, we are able to identify the hypothetical bioactive compounds and therefore design targeted isolation procedure, which really speed up the isolation process. So I will try and show you something about this topic by showing you a couple of examples from our research activity. So the first part of the talk will be dealing with the search for anti-cancer compounds. So the aim of this research is to find anti-cancer compounds, especially against drug-resistant colon cancer cell lines. We actually have a couple of projects that are dealing with this kind of research. One is on leguminous plant species, and the other one is dealing with Asteraceae plant species. But in both cases, we use NMR-based metabolomics. I will be mainly focused on the first one because it was the first project we were starting with this approach. And before moving forward and showing you the results of our studies, I would like to highlight why we decided to focus on colorectal cancer for our research. So here you can see the statistics about the number of cases and the number of deaths. These are from 2020, but the trend is still the same. So colorectal cancer is the third in terms of incidence, and it's the second type of cancer in terms of mortality. I also have to say that recently, in the latest years, there has been a great improvement in the therapeutical approach to colorectal cancer. And in particular, targeted therapy has successfully prolonged the survival for patients. For example, cetuximab, which is a monoclonal antibody that targets the epithelial growth factor receptor, is a very promising drug. Actually, it's currently widely used for colorectal cancer. However, there is a next challenge, which is the resistance to this drug. So there are tumor cell population that are resistant to the cetuximab treatment, and this is usually due to the fact that there are mutations at the intracellular effectors of the epithelial growth factor. But also, even more dramatic is the fact that up to 25% of patients that initially respond to the treatment with cetuximab developed secondary resistance or acquired resistance. And this is due probably to the fact that the initial population is heterogeneous. So it also includes cells that are resistant to cetuximab, and the selective pressure we give to these cells with the treatment leads to the selection of the resistant cells. So even though there are very good therapeutical approach, it's always a good idea to look for alternatives. So the first example I'm going to show was actually a project from our former PhD student, Vittoria Graziani, who was working on 14 leguminous species. So she was studying this plant extract for their metabolite content and the biological activity. And by combining the information deriving from the results of these experiments, she was able to identify a plant extract, and in particular, specific compounds within this plant extract that might be responsible for a very interesting activity. So we knew from the 2D NMR analysis of our plant extract that the compounds were cycloartan saponins. So we were also able to selectively choose the techniques to separate these compounds, and we very rapidly obtained the pure compounds, which were tested for their activity. And one of these cycloartan glycosides showed selective activity against drug-resistant cell lines. And currently there are follow-up studies in progress. We might also have identified the targets, the molecular target, but I won't talk about this part because we still need to validate these findings. However, you can see from this slide that there are several plants which we were testing. One of these was Ononis variegata. You can see here that this Ononis variegata was among the inactive plants. So now you would think, why are you talking about this? This is because we collected another plant from the same genus, Ononis diffusa, and we were studying the metabolic profile of both species. So when I'm talking about the NMR-based metabolomic method, actually I'm talking about a very simple and straightforward extraction procedure. So we usually start with lyophilized plant material. This is directly extracted in deuterated solvents, so solvents that we will use for acquiring the NMR spectra by using ultrasound-assisted extraction. After a centrifugation step, we take the surnatant and this is directly analyzed. For all the studies I will show you here, we use the solvent mixture, a mixture of water and methanol, 1:1, and the water was in particular a buffer, a phosphate buffer, which also included an internal standard. Now, from the comparison of the two NMR profiles, we could observe that there were compounds which were common between the two extracts, but we also had different compounds in Ononis diffusa extract. So we decided to test the extracts from Ononis diffusa for their bioactivity. And what we observed here was that, so here you have two graphs which are reporting the percentage of cell growth on 3 different cell lines. And what you see here is that while Ononis variegata did not show any significant inhibition of cell growth, with Ononis diffusa we observed a good inhibition of the cell growth on these 3 cancer cell lines. We also tested Ononis diffusa extract on cell lines which had acquired resistance to cetuximab, and we still observed activity. I'm not going to discuss in detail the activity because we are talking about preliminary results on the extract. This was an information that we needed in order to decide if it was worth going on with the study of the Ononis diffusa extract. But at this point, I also have to point out that the compounds which were common between the two extracts were also present in comparable amounts. So we could already hypothesize that this compound might be not responsible for the activity. So we already had an important information at our hand. However, Ononis diffusa extract was quite complex extract, so we decided to simplify it by doing a partial purification. So the extract underwent liquid-liquid extraction. We obtained 2 fractions, one in water and the other one in ethyl acetate. The water fraction was further purified on amber light to obtain a water and methanolic fraction. Now, this last water fraction only contained sugars, organic acids, and other so-called primary metabolites. So we tested only the ethyl acetate and the methanolic fraction. So here you have the graph of the activity on one of the cell lines, HT29, which shows this cell line for this test because this is a cell line that is intrinsically resistant to cetuximab. And we could already see how the ethyl acetate extract showed a significant inhibition of the cell growth, which was not shown by the methanolic fraction. From the comparison of the two NMR spectra, we could observe how the compounds which were all already detected in the other Ononis species were all present in the methanolic fraction. And also we observed some signals which were more or less common between the two spectra. And we know from the 2D NMR analysis that these are the signals from structurally related compounds. So from these considerations, we we could focus on the signals that might be responsible for the activity. Of course, that's not so straightforward, it's an hypothesis, but we went on this line. So we saw that from the 2D NMR spectra, we had this substructure. So we had the carboxylic function, which was conjugated with a double bond. And we could also build the rest of the chain here. From the HSPC toxi and the HMBC analysis of the fraction, we could also see that somehow these signals were correlating with signals in this region, which looked like fatty acid signals. So we thought that we were looking at oxylipins. And based on this information, we could once again design a targeted isolation. So the extract was directly separated using a Cephedex-LH20, and we obtained fractions that were enriched in oxylipins that still showed activity. One of these fractions was further purified using preparative TLC, and from the NMR of the pure compound, we could confirm some of the structural information that we already had from the extract information. So, but we could also have more information that was not possible to obtain there. In particular, we could see how the double bond was a cis double bond, so we could define the geometry. And this cis double bond is also justified by the fact that actually we have a delta lactone unit. So from the complete structural elucidation of this compound, we could define this structure, or better, we could almost define the structure, because now we need to work on defining the stereochemistry. So we need to define the absolute configuration of the many chiral centers which are present in this molecule. So now we're working on completing the structural elucidation of this isolated compound, but also on the isolation of the other oxylipins. And we, of course, we will validate the activity once we obtain the pure compounds. How did we use metabolomics in this experiment? Metabolomics was mainly used to follow the activity and to already have structural information on the compounds that might be responsible for this activity. And once we have this preliminary structural information, we were able to design a process to isolate this compound, which was make this isolation procedure much faster compared to the classical phytochemical studies. Now we will move again to plants. This first part of the talk was related to the use of compounds to as possible drug candidates. The second part of the talk will instead deal with plant-plant interactions. And we will start by defining what this kind of interaction is. So we know that plants produce chemicals that can be released into the environment by different routes, and they can affect the seed germination, growth, and performance of other plants, which are defined as receiving plant. Well, actually, this phenomenon is much more complex because we know that also other actors might be involved, like, for example, soil microbes. However, understanding this phenomenon might be very important from the ecological perspective, but also, once again, for possible application, because compounds able to interfere with the growth of other plants might be an alternative to the currently used herbicides. We have some example of this kind of ready of compounds which were isolated from plants which work as herbicides or which were used as lead compounds to produce herbicides. Now, why do we need, once again, alternatives to the currently used herbicides? We know that crops have to compete with weeds, and often weeds are successful in this competition because somehow we selected crops to have better productivity. So we preferred productivity over defense for these plants. So herbicides are an essential tool in agricultural practices, but we also know that they have several drawbacks related to the health environmental issues. But once again, another very compelling issue is the development of resistance mechanisms. And once again, this is related to the use and sometimes overuse of these compounds, which select resistant weeds. So finding compounds which have alternative mode of action compared to the ones that we are currently using might be a good thing to have. However, studying allelopathy is not so easy. And now, a long time ago, during my PhD, I was standardizing a method that is based on NMR-based metabolomics. This method was standardized and applied to a set of Mediterranean plants. And from this preliminary study, we selected a few plant species which showed interesting activity on the receiving plant. And these are the ones shown here: Arbutus sonedo, Scognamiglio, Medicago minima, and Myrtus communis, which were tested on the receiving plant, which is Aegilops geniculata, which is a coexisting species of the donor plant. This metabolomics-based approach basically starts with the extracts of the donor plant, from which we obtain also dilutions and partially purified fractions using SPE, in this case, a reverse phase SPE cartridge. Both the extract and the fractions are characterized using NMR. And then these extracts and fractions are used to treat the receiving plant. The receiving plant is growing in hydroponic conditions, and the compound to be tested, or the extract or the fraction to be tested, is added to the nutrient solution. After a standardized time, we measure the growth parameters of the receiving plant, and they are sampled, they are lyophilized and analyzed by NMR-based metabolomics. So at the end, we are able to see what are the effects on growth and metabolism of the receiving plant species, but also we are able to identify the putative active compounds or mixtures. So once again, we did metabolomics. What I'm showing you here is metabolomics analysis of the leaf of Aegilops geniculata, so the receiving plant, after the treatment with the donor plant extracts. So you can see here control samples, and we can see how on this curve plot along the first principal component, we have a clear separation of all of the treatments from the control. However, we can also observe how we have a grouping here of treatments with different donor plants, which are clearly separated from this other group of treatments. In the first stage, we were focusing on what was happening on these three plant species. So we were performing further experiments, and we were able to identify the allelochemicals, the putative allelochemicals, arbutin, catechin, and other phenolics. The activity was validated on the pure compounds, and we were also able to understand what was going on at the receiving plant point of view. However, it's not on this part I want to focus today, but I want to focus on the other donor plant effects, because I think this best shows how metabolomics can be useful in this case. So we see here that, as I said before, it's grouping away from the other treatments. And the same was also observed when we analyzed the roots of the receiving plant. From the morphological point of view, all of these extracts showed activity on the receiving plant. However, we observed from the multivariate data analysis, we could also highlight which signals in the NMR spectra were responsible for this peculiar behavior. And actually, this was one of the cases where multivariate data analysis was actually not needed. So if we have a look at the spectra, here in this figure, the last one, the blue one, it's Aegilops geniculata control. So it's the leaf of Aegilops geniculata, which is not treated with the donor extract. The middle one, the green one, is once again the leaf of Aegilops geniculata, which has been treated with the donor extract from Daphne. Now, the leaf are never in direct contact with the extract. However, what we observe here in this region of the spectra is that clearly in the plant of Aegilops geniculata treated with Daphne, we have additional signals compared to the control. And with the 2D NMR experiments, we were able to identify the compound which was responsible for these signals, which was daphnetine. So this is a coumarin-like compound, so it would not even be surprising that it has phytotoxic activity. However, if we have a look at the upper part of this figure where we have the extract from Daphne, we can observe that here we have daphnetine, so we have signals from daphnetine, but also a set of signals from compounds which are clearly structurally related. And in particular, we could identify this compound as two glucosides, daphnine and daphnetine 8 glucoside. So we thought that we were lucky. So we were able to identify very fastly the bioactive compound, but of course we needed to validate this finding. So we isolated the compounds. We performed the bioassays at different concentration, including millimolar, which is quite high concentration. It was even higher than the concentration of the compound in the extract. However, after the standardized time that we usually use for these assays, we observed no inhibition of growth or any sign of stress on the receiving plant. So it's like the pure compound was not doing anything to the receiving plant. So while the plant was not stressed, I was getting stressed, and I decided anyway to analyze the leaf from the receiving plant after the treatment. And here we can compare what we observe when we treat with the extract. So here we have the NMR spectrum, and particularly the aromatic region of the leaf of Aegilops geniculata treated with the extract, compared with the treatments with the 3 pure compounds at the highest concentration tested. We did not observe signals from daphnetine or any other of the compounds. However, we decided to go anyway and analyze the roots. And here we observed something that at the beginning looked weird, because while, as you can see here, in the roots of Aegilops geniculata treated with the extract, once again, we mainly have daphnetine. In the other treatments, no matter the initial compound, we obtain a mixture that we could see from the NMR and LCMS analysis, were mixture of derivatives of daphnetine with different glycosylation position, but also number of sugar attached. So we had these complex mixtures. So to sum up what we observed, when we treated the receiving plant with the entire extract, we observed activity. So we observed that the extract was inhibiting the growth of the receiving plant. And we observed the aglycon of the compounds, which were the most abundant compounds in the donor extract, in the root and shoot of the receiving plant. When we use the pure compounds, even at a concentration which was higher than the one of the initial extract, we could not detect any of these compounds in the leaf samples of the receiving receiving plant, but we could observe this complex mixture in the root. So once again, I'm leaving you with an hypothesis, because here we still need to work on this hypothesis I'm going to show you now. But we think that somehow the compounds are deglucosylated, but the glycon is toxic to the receiving plant. However, the plant is able to glycosylate it. And this would not be surprising, because glycosylation is a common mechanism of resistance towards xenobiotics. So in that case, we obtain, it's what we observe here. We obtain a complex mixture of glycosides which are stored in the roots. However, there might be something else in the extract which inhibits this step and then causes the glycon to accumulate and then to have the deleterious effect it has on the receiving plant. We are working on the identification of this component, and it's very likely something that it's also acting at very low concentrations. However, this might be very important because the compounds we might identify could be used also in combination with maybe herbicides, or can be used in herbicide formulations and to inhibit the detoxification process, for example. Or we could use this two-component system made up by the daphnetine and this hypothetical compound as an herbicide formulation. However, the main point I would like to highlight here is that metabolomics here was very important, because normally we would have tested the pure compounds and said, okay, these are not the bioactive compounds, or just hypothesize a synergistic effect. While with metabolomics, we could observe that something different is going on when we use the entire extract and when we use the pure compounds. So with this, I would like to thank not only the people directly involved in these projects, but also my current research group and all the research groups I had the chance to work with, the funding agencies, and all of you for your attention. And I will be happy to take any question.
Thank you, Monica. Luke had to leave, but if you have any questions, please feel free to unmute yourself and ask questions or put them in the chat.
Hi, Monica. I was wondering about, this is Stacy. I was wondering about the oxylipids. I've never heard of them before. And I was wondering if you know where they are localized in the plant. Are they sort of a cell wall thing, or is it like a cuticle lipid? Do you have any sense for that?
So far, I do not have this information. So I'm actually curious to understand more also about both the localization, but also the roles in plants, because especially in this plant, they are really abundant. So they are a very big component of the extract.
That's super interesting. It'll be fascinating if you figure out where they are and what they're doing.
Yeah, that's next step. Because as I said at the beginning, so this project is now dealing with the anti-cancer activity, but I'm really, really curious to understand why these compounds are produced by this plant, why they are so abundant, what's the role, and where they are localized, of course.
I had a question. Can you elaborate a little bit on the typical use cases for NMR-based metabolomics versus LCMS, and what situations you'd use them in?
Sorry, can you repeat?
NMR-based metabolomics versus using LCMS-based methods. I know one is NMR, you get the structure directly, but LCMS also gives you more compounds. So in what cases would you use?
Yeah, that's a very, let's say, common and old question, and also interesting question, because, well, I think that the best thing would be to have the chance to do both, because as you said, with NMR, you have structural information, you can be quite confident about the compounds. I mean, you really see the compounds somehow. It's very rarely that you are not able to at least understand which class of compounds you are dealing with. But on the other hand, LCMS is much more sensitive, so you can see a lot. So I think that when you have to choose, because sometimes you do not have the chance to do both, I would use NMR-based metabolomics for this preliminary assessment, because it tells you something about all the kind of metabolites that you can extract with your solvent, because we also have to take into account that we include a bias with the solvent choice. And then once you already have some hypotheses, I would go for LCMS, but with more targeted approach. I wouldn't do unbiased, untargeted LCMS for this kind of studies. I mean, I would use NMR-based metabolomics to guide also the LC-MS-based part. But people that do LC-MS-based metabolomics would probably tell you something different. It's just my opinion.
Thank you. Yang Yi?
Yes, thank you, Monica, for the talk. And I have a question regarding the NMR. So I wonder what kind of purity you're expecting or is acceptable for doing the biological studies. And since those kind of nature metabolites are really, really complicated. So it's really struggling to get a purified compounds. So I wonder what's your opinion?
Yeah, so when we're talking about the biological testing on the pure compounds, I would always have a compound which has also been purified, for example, on HPLC. So I would go for a high purity of the compound.
How do you judge your purity?
Once you do your HPLC, you're able to see if there are other compounds there. Of course, there might be compounds that are under the limit of detection.
That is not necessarily true. I think the best way, maybe more common way, just using NMR, like proton NMR. I think most researchers using proton NMR to check the purity. So I wonder, in your case, what is the percentage of the purity in your extraction? Thank you.
Yeah, well, I do not have this information yet, because I showed you the pure compound, but this is we just did the structural elucidation, so we didn't test yet the pure compound there. Mm-hmm. So for this one, I cannot answer yet.
Okay, that's fine. Thank you.
Thank you.
Hi. First of all, thanks, of course. I just wanted to ask if the slides will be available somewhere.
Sorry, I didn't.
Oh, I'm sorry. I just wanted to ask if the slides will be available somewhere, because I want to look into it again.
I think it will be uploaded to the...
Yes, this is being recorded and it will be uploaded to YouTube.
Oh, thanks. Perfect.
Okay. Michael, go ahead.
There we go. Yeah, thanks, Monica, for the talk. So you mentioned that near the end on your hypothesis of this allelopathic project, that you expect that the plant is glycosylating it in some manner. So, and this is based off of also observations of like higher molecular masses, perhaps in that wonderful indicative plus 162 signals on LC.
Yeah.
something like this as well? And do you have any information on how many extra sugars are being added? Is it just the same compounds, like the 7 or 8 glycoside, or is it?
Yeah, you have also compounds where you have like 2 units, 2 exos units bound to 1 carbon or to 1 of the OH, or you have a very complex mixture.
Okay, so that's
So
cool.
it's like glycosylating
It's
everywhere.
polyglycosylating, right? Serial glycosylation. So I mean, I might have some exposure to serial glycosylation in my work and in the past. So one other thing that I found interesting is that you also purported that perhaps it's a inhibition of the activity which glycosylates it, which might actually help bolster the growth inhibition of these compounds. So I don't know, on one of your slides you had some of the compounds that you see there. Maybe it was a picture of myricetin or some sort of flavonoid or something like this. Actually, I don't know what was on that slide, but there's a lot of different flavonoids that have been shown to have pretty good alpha glucosidase inhibitory function, and some of these glycosidases obviously have reversible functionality in being glycosylators, and serial glycosylation is one of their functions. So that's a good theory, and I would definitely go ahead and look forward into that. I was just saying that it's good, it's a good hypothesis. So, and perhaps combining the compounds which are maybe the flavonoid extract, if you have some sort of portion which is coming out on your SPE that's mainly flavonoids, that would be a really cool thing to test for combining the activity of these 2 fractions.
Yeah, that's a good point. However, I was not showing here, but I also can tell you that it might be not flavonoids in this case, the active ones, because I already have some fractions I was analyzing. It looks like more kind of terpenoidic compounds.
Okay.
But I need still to validate this, so that's why I was not showing you this part.
Okay, so you've already started
Anyway,
combining these fractions to see. Cool.
Yeah.
That's good. Cool.
Thank you for your comment anyway.
Yeah, thanks.
Any more questions? If not, then let's thank Monica for the wonderful talk and for answering the questions. And if are you able to stay for a few more minutes? Okay. Yeah. So if anybody wants to have an informal chat for the next few minutes, feel free to stay. Otherwise, thank you all for attending.